Okay, so we need to talk about cell cycle and mitosis, and just so that everybody gets to hear their favorite song that has to do with this.
Are never, ever, ever getting back together. We are never, ever, ever getting back together. You go talk to your friends, talk to my friends, talk to me. But we are never, ever, ever, ever getting back together.
You're welcome. All right, so we need to talk about DNA first. First of all, a lot of the stuff we know already. All the genetic information is stored in our DNA, and they're divided. All the units of information are called genes. We know that, and they're all passed on down to our descendants, and other animals and plants all do that to theirs too. Now, when the cell divides, we need to make sure that each daughter cell gets exactly the same DNA that the parent had. That way, everything keeps moving along. DNA is packaged with proteins to form chromosomes. We've seen those in class already. Each parent cell will give one copy of every single chromosome to each of the new daughter cells. That's another term we know already from before, during mitosis in this thing. Meiosis is something we'll talk about next time. That actually reduces the number of chromosomes in half. So don't worry about that right now, but we will talk about that a lot in the next chapter. So this section, what we need to learn is the importance or the significance of chromosomes in terms of what they have, and how DNA is packed into cells. So when a cell is not dividing, chromosomes are present in the unraveled form. Remember, it kind of looked like spaghetti. They were in that spaghetti form inside the nucleus. So the nucleus would be here, and then we kind of drew it like this. It was all squiggly inside there. During cell division, these fibers condense into these dreadlock-like things that we showed before. Okay, this one, chromatin, is this one. That's very, very important. When it talks about chromosomes or chromatids, those are that form right there. Okay, so in eukaryotic cells, DNA is packed into different number of chromosomes. It depends upon what species you are. The way they're packed, there's proteins that are involved. Remember, DNA is nucleic acid. That was one type of molecule we talked about before. Proteins is another one. The proteins that DNA is wrapped around are called histones, and they form things called nucleosomes. You can look in page 139 to see what all that stuff kind of looks like. Don't worry about the number of base pairs wrapped around 8 histone molecules. You just need to know that histones are the proteins that DNA gets wrapped around. Here's a picture also. What's important about these is that if this is a gene right here, if this is right here a gene we need to have expressed and it's wrapped around that protein, it can't get expressed very easy. It can't get accessed. So it kind of shuts off those genes, and that's what this screen is kind of showing here. Now if the gene's over here, it's no big deal. It can easily be accessed and activated. Okay, so chromosome number and content. So every species has a different number of chromosomes. For us, it's important to know we have 46 chromosomes. You get 23 from mom, 23 from dad, and that adds up to 46. Roundworms have 2, and then some plants called ferns have 1,000. Crazy. The cell cycle and mitosis. What we need to learn here is we need to be able to identify the stages of the cell cycle, and we need to know the structure of duplicated chromosomes, which we've kind of gone over all of this stuff. Excuse me. The sister chromatids we've talked about, centromeres we've talked about briefly, and kinetochores, those you've seen once, and we'll see more of those. The significance of mitosis, why it's important, and be able to describe the process. Now, we've been working on that. That shouldn't be very. So when cells reach a certain size, they need to either stop growing or divide, and you can see that on page 137. If they just get too big, there's not enough surface area for the amount of volume that they have, so they have to divide or just stop. So the cell cycle, we talked about, it's the stages that the cell goes through from between cell divisions. Right? We know the G1, S, G2. We've done all that stuff. Those G1, S, and G2 are interphase, and then the M phase is the division part. Most of the cell's life is spent in interphase. That's when it's doing its normal job. We've seen that already. It's when it's metabolically active. That means it's making molecules, it's making proteins, lipids, other important molecules, and it's growing. And we've said before, it's divided into G1, S, and G2. So G1 is between the end of mitosis and the beginning of the S phase. It's normal operation. Stuff's happening. We've talked about this. This should be easy. Synthesis. That's when the DNA copies itself. This is the very, very important part. At some point during G1, the cell decides, okay, I'm going to divide, which means it needs to go and copy its DNA. After it copies the DNA, it's going to make more proteins, make lots of proteins, and get ready for the M phase, which is mitosis. It really, a lot of times we'll call mitosis the whole thing, but really mitosis is just the division of the nucleus and the splitting of the chromosomes. And that's the one that's divided into the 5 phases, the prophase, metaprophase, or sometimes you'll see late prophase. Prophase, that's another possibility. Metaphase, anaphase, and telophase. Your book doesn't use prometaphase, but that's okay. A lot of times, cytokinesis, or the dividing of the cell, gets thrown in there, but really it's a separate part of the M phase. So really, M phase isn't just mitosis, it's both mitosis and cytokinesis. So prophase, we know most of these things, so it shouldn't be very, very difficult. We've said that the first thing happens is the chromosomes have to get compact, right? They can't be that chromatin form because they will break when they try to separate. So we need to make them in some compact form that they can separate. And when they do that, after they're duplicated anyways, they form sister chromatids. So this is one chromatid, and this is its sister. You could do it the other way, say this is one chromatid, and this is its sister, right? Just like you and your sister if you have a sister. Sister chromatids are held together at the centromere. That's this part right here. This kinetochore part right here is part of the centromere, and that's what grabs onto the microtubules of the spindle fiber, which helps divide them when it gets time to do that. Each of the pole has something called a microtubule organizing center, which we've so far just called, we've called them centrioles. Centrioles right there. The whole thing put together is called an organizing center. Centrioles are just a part of it, but it's the big part for us, so that's all we really worry about. And then there are these tubes coming out, microtubules, or the spindle fiber. Another name is mitotic spindle, same thing. Spindle fibers, mitotic spindle, same stuff. So towards the end of prophase and before the beginning of metaphase, the nucleus starts to dissolve and go away. The kinetochores, remember that's the part right here inside there at the centromere, right? That's right inside there. I didn't want to do that, but that's okay. Right in here, they capture some of these microtubules. Right? They capture these. You'll see some of these microtubules. You'll see this in a second. I'll explain these. But there are some microtubules that don't catch chromosomes. They catch other microtubules. And then there are other ones that just sit around here like this. At this point, this is when the chromosomes start to move towards the center of the cell and line up on what's called the midplane, or the sometimes called the metaphase plate, different names for it. Like I said, the microtubules, they're the polar kind. The polar ones are the ones that went and found each other. So if this is a microtubule center over here, and this is one, and this one go over here, these are the ones that catch like this. Those are the polar ones. The kinetochore ones are the ones that go to the middle to a kinetochore. Right? So if they're over here, they'll go and attach to the kinetochore. And then the astral ones are the ones that are over here at the center, and they kind of don't get very big. They just kind of hang out here, and they help make sure that these parts, get to where they need to be. So it helps move the centrioles. Okay. Metaphase, easy one. This is where they all line up. Hopefully this is the one everybody knows. The chromatids are completely condensed and they're nice and lined up to get ready for division. Anaphase is where they start to split apart. You can see the X's are no longer X's. They're only half of the X now because they break apart right at the centromeres. And they start to move towards opposite poles or opposite ends of the cell. Telophase, they finally get to where they're supposed to be. They get over here at each end. They kind of start to uncoil a little bit. The new nucleus starts to be formed, and all these spindle fibers disappear. And you can actually start seeing the nucleoli sometimes. Cytokinesis is when the cytoplasm actually divides. Remember we said it's a little bit different than mitosis, but it often gets lumped in there together. So the cells actually divide into two cells, and that's called cytokinesis. Animal cells, remember, makes that cleavage furrow. We talked about that the very first day back. Plant cells are a little bit different. Remember, they can't pinch off in here, so they have to build their own wall. It's called a cell plate. We talked about that. That was pretty intuitive, too. I think you guys figured that out really easily. Then the daughter cells. What's important about mitosis is that each of the daughter cells has exactly the same number and kinds of chromosomes. That means these things are genetically identical. And that's important for this type of that's a horrible writing thing right there, but that's okay. That's important because we want these cells to be exactly the same. We don't want to start mutating our cells once we're already alive. We want them to be genetically identical. Normally every cell has the same genetic makeup, which is what I just said right there. The organelles get divided so that each one has enough to each daughter cell can survive, enough mitochondria, enough chloroplasts, whatever it needs. Now these have their own DNA, which is kind of interesting, and they divide when those new ones are made. And that's it. We're all done.